So thank you very much for joining me for my talk tonight. I'll be talking about seeing the epidemic through the trees, or why sequencing is important for COVID-19. So in English, we have an expression, you can't see the forest for the trees. And what this means is that someone is so caught up in the detail that they can't see the bigger picture. They're missing the forest because they're too busy looking at the trees. Now, a lot of you might be watching this right now from some type of quarantine, lockdown, or social distancing. Every day the case counts are rising and the forecasts aren't looking great while we wait to see if the implementations we've put in place are enough to stop the pandemic. And then there's people like me who are calling out for more sequencing. Let's do more sequencing. Isn't this just a detail? I mean, is this really important right now? Or are scientists getting caught up? Are they missing the forest for the trees and getting caught up in the details of sequencing when they should be focusing on something else? Well, perhaps not surprisingly, I would argue that no, we're not. And that actually we can see the forest through the trees. Now, of course, I'm not talking about the trees that grow outside. I'm talking about what we call phylogenetic trees, which we make from the genetic material of an organism. And I would argue that we can use these phylogenetic trees to actually better understand our forest or our epidemic. So first of all, what is a phylogenetic tree? What's phylogenetics? Well, this is a way to reconstruct and record relationships between individuals or species. So these are kind of like family trees for different animals or organisms, except for we use the genetic material to find out what they are, rather than them being written down, which is handy because viruses and animals don't write a lot down. So how do we use this? Well, some of you might already know me, and if you do, it's likely that you might know me in association with NextStrain. This is a really cool website platform where we track the coronavirus in real time around the world and through space. And we actually do this using phylogenetics. So the big tree that you see that has dots and lines is a phylogenetic tree. And this is what we use for all of our analysis and research and to create the maps and animations that you see. So let's go back to the beginning. How do we do this? So we do this by extracting the genetic material from the virus. This is RNA. It's kind of like a virus version of DNA. And we can convert this into the familiar string of ACTG that you probably remember from school. When we do this for a lot of viruses, we end up with a lot of sequences, a lot of genetic material. And these are what we use to track the virus. And we do this by looking at these little changes, like this one, these mutations, that show us which sequences are more closely related to each other and which ones are more different. Now, it's worth taking a minute to talk about mutation because we tend to think of this in kind of the superhero action movie context. Oh no, the virus has mutated, it's getting much worse. But as is often the case, reality is a little more boring, but also safer. So let's talk about mutations. Let's take an example where I have a manuscript or a document that I want to copy a lot of times. Then I'm going to pass it to you and you'll copy it a bunch of times. You can imagine that somewhere in this copying, I'll hit the wrong key and I'll make a typo. And then when you get my copy, you'll think that typo was part of the original, so you'll continue to copy that typo as well. But you also might start introducing your own typos. And you can imagine that as this manuscript gets passed on and copied, there will be more and more typos introduced. This is the same for viruses. Every time they copy, and they have to copy themselves a lot of time, there's a chance that they'll just make a typo. Now, to go back to the document example, the great thing about these typos is we can use them to trace the lineage of these documents. So all the ones that have the red typo, they came from an ancestor with the red typo, whereas those that had the green typo, they have an ancestor that had the green typo. And you can see how we can recreate how these different documents got copied and passed on over time. It's exactly the same with viruses. They make these typos, and these are what actually allow us to track the virus through time. Now, what's important to consider is that if I gave you some of these documents and they had some typos in them, you'd probably still be able to read them. They would still make sense. You'd still be able to get the general gist of the document. And this is true for viruses as well. Most of the mutations that happen don't change the virus function or anything else about the virus. The next most common thing that's likely to happen is actually that the typos change some really important word in the document, or there's just too many typos and you can't read it anymore, so you throw it out. This is the same for viruses. Most mutations are harmless, they don't change the virus, but it's much easier to break a gene or a protein, and this breaks the virus. So there will also be mutations that are bad, and these are ones that stop the virus from functioning. They won't go on to replicate and infect everyone, so they get taken out of the gene pool. So most mutations are harmless, but these are what allow us to track the virus, and a few will be bad, but they'll be taken out of the gene pool. So how do we use this in biology? So when we take these genetic sequences, here I'm not showing them as ATCG, but I'm showing them as just lines where the differences are different colors of dots. And as I said, these are the typos that viruses make as they replicate. And we can look at these different dots, these different mutations, and we can see how they might be related. So all of these sequences have a blue mutation, so they must have come from an ancestor that had a blue mutation as well. But then only some of them have a yellow mutation and only some of them have a green mutation. We can draw a really simple tree that shows us how these sequences are related. So they all come from an ancestor with a blue mutation, but then only some of them have the yellow mutation and only a couple of those have the green mutation. So while A and B are identical, they have exactly the same mutations, they then have a distant cousin, C, who doesn't have the green mutation but has a red mutation. And this is exactly the process that we use with our real sequences from the coronavirus to see which ones are more similar and which ones are further away. Now, just quickly, I want to talk about some of the parts of a tree so that I'm using the same language that you'll understand. So our samples that are at the very end, we call the tips of a tree. And then the branches that are this way, just like a real tree, we call them branches. And where they meet, we call internal nodes. And these are hypothetical ancestors. So we know that a virus must have existed that gave rise to sequence 1 and sequence 2. We probably haven't sampled it, but we can infer that it must have existed and we can make some assumptions about what that might mean, which I'll talk about a bit more later. Now, one thing that's important is that it's these horizontal branches that tell us the genetic differences between the viruses. So the difference between sequence 1 and sequence 2 is the length of this branch and the length of this branch. The difference between sequence 1 and sequence 4 is this branch, this branch, this branch, and this branch. So you can see that sequence 1 and 2 are more closely related than sequence 1 and 4. The horizontal lines like this just show us connections. They don't have any scale. So they're just showing us how the sequences are connected into a diagram. So how does all of this apply to pathogens? So when a pathogen moves through a population, this is represented here with these red circles that are infected people, and they go on and they can either infect one person or a few people, or sometimes they don't infect any people. And then how long they're infectious is shown by these horizontal lines, and then where they stop being infectious is shown by these little stopping points. As a pathogen moves through the population, it'll start acquiring mutations. And these mutations are what we're going to use to track it. But of course, we're usually not lucky enough to have a sample from everyone with the disease in a population. We usually just sample some small number of those people. So here I've shown that with the blue dots. So the blue dots are when in an infection someone gets sampled. And these are the only things that we're left to work with. So here we're going to take out the red dots, and this is what we're left with, just our sampled population. However, we have the mutations that the virus created while it was replicating, and we can use these to reconstruct a kind of vague history, an overall picture of how this virus must have spread through the population from the samples that we have. And this is what we're able to do to track the coronavirus in real time. So, how do we actually use this data? There's a couple different ways we can do this, and I don't have time to go into all of them, but I'll talk about one really good example. So one thing we can do is we can infer things like location. So if we know that our samples were taken, for example, 2 of them were taken in Europe and 2 were taken in North America, then we might be able to say, okay, if these 2 were in Europe, then this hypothetical ancestor was probably also in Europe, whereas the hypothetical ancestor of the deeper nodes might have been in North America. And we can use inference to draw a picture on the tree of where we think the virus was going back in time. And this can be really useful, but it's also got a lot of pitfalls. So I'm going to use a couple of examples from Dr. Nicola Muller, who's in the Bedford lab, also part of the Next Strain team, to show you what I mean. So let's say that we have some samples from an orange country and some samples from a blue country. Because of one particular node, one particular sample, this one here in orange, we might make the following inference. We might think, well, the top of the tree has more orange samples, the bottom has more blue samples. So the virus must have been circulating in the orange country, and then it looks like it came into the blue country 3 separate times. There were 3 introductions to the blue country. But let's imagine for a second that we don't have this orange sequence, that we only have the other orange sequences. Now we might make quite a different inference. We might say, well, the top of the tree seems to be orange, the bottom is blue, and so maybe there was just one introduction. It was circulating in the orange country, and then it moved into the blue country just once and spread onwards. But let's take it a step further. What if we only had one orange sequence? For whatever reason, we're not very good at getting sequences from the orange country, so we only have one. And here it is. Now we might say something completely different. We might say, okay, well, this virus is obviously really circulating in the blue country, and it looks like in the past it had one introduction into the orange country. So this are 3 very different inferences that we've made, just depending on the samples that we had. Now, I've shown this to you going from left to right, but in reality, the data actually moves in the other direction. We usually start out with less data, like right now, particularly early in an epidemic, and we move towards having more data. So we need to be really careful that the inferences we make now are ones that we feel like we have enough data to make, or else when more data appears, they might be proven wrong. And even though that's just the way science works, we need to be conscious of the fact that these can have real life implications. So if we say that a virus spread from one country to another country, people can get really upset at the other country for not doing more to stop the spread. Or if people believe that one person led to an outbreak or led to the outbreak in a country, this can have real life hurting consequences for that person. So we need to be very careful about the inferences we make and that we can support them. But what does this actually mean for the coronavirus? So I'm going to move now and hopefully switch screens to next strain and show you with the live site what we see with the coronavirus data. So this is the live site where we have over 1,000 sequences now, and you can see that they've come from all over the world. And this data set is really diverse. We have data going back as far as the end of December, and we have data from as recently as just a few days ago in March. And we've colored this by different regions around the world. So you can see that the Americas are mostly red, Europe's kind of orange and green, and Asia is turquoise and purple, and Australasia is turquoise and purple. And I'd just like to point out a couple of examples. So first of all, we can zoom in on kind of a famous story. And I'm going to switch the map into what we call divergence mode. Sorry, it's behind my screen. And all this means is that instead of looking at time on the x-axis, we're going to look at number of mutations. And I'm showing it in this mode on purpose because time is another thing that we can have to be really careful about the inferences we make. We can make a tree that makes it look like something happened at a certain point in time, but we may not have the confidence to actually say that right now. So I'm going to show this in mutation view because these we can actually count from the sequences and they don't necessarily say anything about when something happened. But what's interesting about this cluster in Washington is that these samples, a lot of them were taken at the end of February, and they were actually part of a flu surveillance study. So people just signed up and got a kit, and as soon as they were able to, as soon as they got symptoms for anything flu-like, they took a swab and they sent it off to be sampled. And this helps us get an idea of how flu spreads around the world. But the Seattle flu team had the great idea of starting to test these for coronavirus because they're random samples from a population that isn't sick enough to go to a doctor. What they found when they did this was that yes, we did have coronavirus samples, and all of those samples from Washington clustered really closely together. Now, when we first looked at this, this cluster wasn't as big as it is now, but what it showed us was that there was significant undetected circulation of coronavirus in the general population in Seattle. And this is really important because we can distinguish between whether, for example, some people had been elsewhere in the world, gotten infected separately, and then returned to Seattle, which might lead to one kind of intervention, from the situation that we actually saw, which is the sequences were very close to each other. This virus was actually spreading in Seattle. So we probably need to start implementing things like ramping up testing and social distancing. Now this cluster, as you can see, has grown really, really big and also includes some other states in America, though we can't necessarily infer directionality at the moment of which states maybe had the virus first. But what it does show us is that this virus has been circulating in the US and particularly in Seattle for quite some time. Zooming away from that, I can take us into, my computer's struggling with the recording and showing next strain, but I can take us into this really big European cluster, and I'll zoom in a little on the map as well. And here, we have to be careful with what we say because we have very different sampling available for different countries. So there's quite a lot of samples available in the UK, in the Netherlands, Belgium, France, and Switzerland, but not so many from other countries, and that will skew our viewpoint. It doesn't mean that those countries don't have plenty of cases, it just means we don't have sequences there. But what we can see really clearly is that the lines connecting these European countries go absolutely everywhere. And this was true, I think we all know that it's true now, but this was true even weeks ago. We knew that this virus had spread extremely well across Europe. So the measures of borders closing that happened a few weeks ago probably didn't do anything at all for stopping this virus from getting to different countries. It was too late. This virus had already spread throughout Europe. It can spread easily because you can have not very strong symptoms or be asymptomatic, or if you do develop symptoms, you can spread it before you have those symptoms. And so people just traveling and working as part of their daily lives were spreading this around Europe very effectively. And what matters now, and what mattered a few weeks ago, is stopping transmission between people, not necessarily between countries. And in this cluster, we actually have plenty of other samples from Georgia, from the US, from Australia, and even from, I think we have a few down here from Hungary, and even from Vietnam. So even this quite tight kind of primarily European cluster has worldwide connections. This virus really has gotten everywhere. And that happened a few weeks ago. This isn't a recent development. One other thing that I'd like to show while we're zoomed in on Italy, or while we're zoomed in on Europe, is an Italy story. So if I scroll down very slowly, my poor computer is struggling, we can filter our samples so we just show our 4 Italy sequences. And it's a little bit hard to see at the moment, but essentially we've got 2 up here at the top, and we've got 2 down here. Now, we can't say a lot with just 4 sequences from a country. I think that we're hopefully going to get some more soon. But what we can show is that these sequences do not cluster together. So if you remember the Washington sample, those were really close together. These are not. They're in completely separate parts of the tree. And what we can learn from this is that it's likely that there were at least 2 separate introductions into Italy. And this is important because it tells us if there was just one introduction, we could tell ourselves maybe there's something we could have done to prevent this. Maybe if we just stopped that one introduction, then this wouldn't have happened. But when there's 2 and probably more, it tells us that stopping this would have been really difficult. Particularly remembering what we knew about the virus and its spread at the time, we would have had to have completely different measures to even have a chance of keeping this virus out. And this is important because it changes our view of how this started spreading in Europe and whether there was something that we might have been able to do to prevent it, apart from really rolling out quite strict measures a lot earlier. So the last thing I'd just quickly like to touch on, because I'm aware that I don't want this to be too long for everyone to watch. So if I can zoom back out again on the map, I'll take you to a cluster down here, which is quite diverse. Well, yeah. And it includes samples from all over the world. So Canada, England, Australia, I think we've got some from New Zealand, from Georgia, from Kuwait. And what's interesting about these samples is that even very early in the epidemic when we got them, they clustered really closely together, even though these people don't know each other at all. And what we found out was that we know the travel history for some of them. So for some of these Canadian ones, we knew that they'd been to Iran. And this is really important because what brought these different samples together, even though they were from different places in the world, was their shared similarity in sequences and their shared travel history to Iran. So we can say with quite a lot of confidence that these people weren't infected before they went or after they came back or in a layover somewhere in the world. They were infected in Iran. And what we can also say from this is that the other samples in this cluster were also probably infected in Iran or by someone who's been to Iran. And we can say all of this without actually having any Iranian sequences. We don't have any Iranian sequences in our build yet, but we can still make inferences about where the virus came from and how it spread. So we'll go back to the main presentation now, just to wrap up. So I started out by talking about whether we're missing the forest for the trees. And I hope that I've convinced you that sequencing is actually really powerful in these instances, and we can use these phylogenetic trees to better understand our forest or our epidemic. Sequencing is really powerful. We can understand things like whether transmission is local, as we see in Washington, or whether it's imported from different places, and the sequences, even though they're sampled in the same place, come from a really diverse background. And in the future, when we have more data, we'll actually be able to start estimating when these importations may have happened and how many there might have been. We can also look at the big European cluster as an example of how the sequences are connected. And this shows us that the virus is moving well across Europe, but also around the world. It's not being stuck in small pockets, it's actually transmitting globally and between countries without an issue. We can also use it to try and understand how many introductions have occurred, like in the case of Italy, where we see that there's at least 2 separate introductions, and this can inform us about what may or may not have worked as far as different kinds of things that we may have put in place to try and stop the virus, or whether this was really something that probably was going to happen over a matter of time. And then finally, we can use it to try and understand how disconnected samples relate to each other, and whether transmission happened in the same place, like the Iranian example. Finally, I didn't have any time to talk about it in this talk, but we can also use it for things like understanding where this virus came from, so when it jumped into humans, and where it might have come from, what animal host it might have been in before. At the moment, for example, we see there are really similar sequences in pangolins and in bats. And as we study this further, we'll probably be able to better understand how it was able to jump into humans, what differences had to occur, and which viruses that circulate in other animals might be most closely related to this coronavirus. But as I hope I made clear during my talk, we have to be cautious. Particularly during an epidemic, we don't have much data, we're moving very quickly, and it's very early in the epidemic. Some things are hard to estimate with confidence, but if we do them wrong, if we're overconfident in our estimates, they can have real-life consequences for countries and people. But sequencing in general is incredibly powerful, and we can use it to inform our responses, to make sure that we're acting appropriately for the scenarios that we see reflected in the genetic material of the virus, as long as we remain aware of our limitations. And with that, I definitely could not do this on my own. I'm part of an amazing team, the Next Strain team, which is spread between mostly Seattle. There's 2 of us here in Switzerland, Richard Nair and myself, and then James Hadfield is in New Zealand. And all of us are very happy to work together, and we make Next Strain possible, along with, at the moment, many, many kind volunteers and developers who are donating time to keep Next Strain running and make it even better. And with that, I'll end my talk, and I'm very happy to take questions.